All right, great. So another question that came up, and this was Paul who asked, how does a virus hide and cause no disease, but then all of a sudden causes disease, such as herpes? So how does that work? And how common is it actually?
So this is getting booted to me, I think, as the resident virologist. We consider generally three types of infections. We have these acute infections where a virus comes in, there's an immune response, there's often some disease, and there's transmission, and then you no longer have that virus physically in your body. Okay, so that's an acute type of infection. And then on the other side of this, we have latent infections. And herpes viruses and papillomaviruses are really good examples of viruses that can maintain some sort of latency. And so let me talk about latency, and then we're going to come to a third class that we call persistent. So the latent viruses encode a number of genes that actually prevent them from replicating. So in the case of a herpes virus is going to get into an epithelial cell, it's going to migrate up neuron bodies. This is herpes simplex, they don't all replicate in neurons. And then it's going to sit in the neuron, and it's not going to reproduce. Okay? Many of its genes are activated to prevent it from replicating. And so latency is actually an active state in this circumstance. Okay? But there's no expression of the viral genes that would make the proteins that our immune system recognizes. And so there's unlikely to be an immune response in a latent infection. Papillomaviruses, these cause cervical cancer and also cause warts. Have a slightly less sophisticated, it's a pretty novel mechanism, which I encourage you, there's some nice information on the web. It's a smaller virus, fewer genes, same concept. It's in your skin, you know what a wart looks like. There's no expression of those proteins, so there's no immune response until the virus is ready to be transmitted. And across the animal kingdom, there's quite a few examples of viruses that have the ability to control the replication cycle to just a burst that's associated with transmission. But this more complicated and intermediate stage is persistent. And this is where the virus itself doesn't really have a mechanism to shut itself off and to hang around. The host immune system can't quite get rid of it. And the virus has some mechanisms to stay with cells. And HIV is probably the best example of a persistent virus infection that by the nature of the cells that the virus infects, those cells are long-lived, and if you can't get rid of the virus until you get rid of the cells. We're learning a lot more about persistent virus infections these days because we have more sophisticated tools to look for low levels. So the difference, I think, the latency, again, is an active event, and the virus has to have genes encoded to sustain that. The acute viruses often don't have really good ways of overcoming host immunity, so they get in and out really fast. And then the majority of viruses can have these persistent life cycles because biology is imperfect, right? Not everything, our immune system isn't capable of getting rid of all viruses. And so some of them will hang around for a bit longer.
I guess it does speak to, more broadly speak to Andrew, whose wonderfully animated movie on parasite adaptations. Almost all infectious diseases, infectious agents, has got an enormous repertoire of tools that they use to try to neutralize all the tools that we have to try to kick them out of the system. So in viruses, you can classify them in maybe a slightly smaller number of different types of tools they employ. So then when you look at bacteria or worms or something, then they got a large number of other things to try to avoid being evicted from those.
Well, do we want to pick up on that? Because there was this general question about coevolution. So what's the sense? I mean, are these different strategies basically just different responses to different evolutionary pressures?
Well, I think, well, yes, in part, and I guess following from Mary's comment about diversity, nothing is perfect. There's lots of things are starting in different places, and they have lots of different tools available to them. So, for example, a worm, which can be quite large, getting up to fairly big chunks of a metre, maybe as thick as your finger even. That's like a big foreign object in your body. It's like an organ transplant. And they can secrete huge amounts of substances because they're so big. And those substances then make possible all sorts of interference with immunity. Obviously, that's not such an option for a virus. It hasn't got the capacity to secrete huge amounts of substance like that. And so it's got to go under other tricks, antigenic variation and shifts and drifts and things. Yeah, I mean, from my point of view, I think the whole thing is hard not to see it as a very strong co-evolutionary pressure. The tricky part is to identify the specific pressures, but that it is a result of co-evolution seems pretty clear.
But I think it's also important to remember there's a whole community of parasites,
yes.
so that when your worm is doing that, it may let something else in, may let a bacterium in, so that can sort of increase. So it's this interaction between all the parasites in the community.
Yeah, I think that's what I mean about pointing your finger on what is actually going on.
Yeah.
It's very hard to know which particular parasite trait is a consequence of which particular host trait in many situations because of that. The great diversity of things going on. So and every new defence tool, just like a real arms race, every new defence tool is a new way to get in and interfere with things.
Yeah. And the consequences of that can actually be really very complex, right? In the sense that there are quite a few worms that can effectively instigate a chemical warfare by turning on aspects of the immune system that are deleterious to other worms that might be competing for resources within the gut. And so you actually have the use of the immune system, the use of the host immune system by one parasite against another parasite, right? And there's lots of inherent trade-offs in our ability to fight off infection, right? Whereby either the upregulation for one parasite either has a positive or a negative consequence for all the other things that are infecting us at any given point.
If we each wrote down all the infections we'd had during our life, they would look totally different. Everybody would be totally different in what they'd had and what their immune responses had been over the integral of their whole life.
I think another interesting aspect of the polymicrobial thing is that now with genomics, bacterial genomics, you're suddenly realizing that bacteria will exchange whole called virulence islands, a whole great big segments of genes that they will share with each other and that are specifically designed to overcome host defenses.
I think one of the best examples of coevolution that comes with viruses, and again, remember a virus is an obligate intracellular parasite, so they must be quite well adapted to use proteins and genetic information from their host. And so we can see the exchange of proteins in the host environment here too. Smallpox is probably the best example where there are proteins, they're mimics, right? The virus has a protein that mimics one that's a host protein. And as the virus evolves to outcompete the host protein, we can see over time that host protein has also evolved. So do that with your worms.
You'd be surprised. You'd be surprised.